So the next type of transport is active transport, which is helpful to think as the opposite of diffusion. So if diffusion is moving things from a high concentration to an area of low concentration and does not require energy, active transport is the reverse of that. So in active transport, you are moving molecules from an area of lower concentration to areas of higher concentration. So now you're going up that gradient, which is like walking up a hill, and it does require energy, or it does require ATP. And typically it does also involve the assistance of protein carriers. But for a cell to engage in active transport, it would want to move things from an area of low concentration to high concentration. So we're going here, we're going from low concentration to areas of higher concentration. And again, that does depend on energy, because you kind of have to walk up that gradient, low to high, go up that hill. An example of active transport is actually endocytosis. So endocytosis is essentially cellular eating. So it would have to engulf things such as food to use as nutrients, or even if we're thinking about something like a phagocytic cell, it would engulf something like a bacterial pathogen in order to destroy that bacterial pathogen. But that action of engulfing something and internalizing something into a cell is consistent with active transport. So with endocytosis, it does require energy because the cell essentially has to extend its cell membrane outward and then engulf those food particles, liquid particles, and even potentially microorganisms if we're talking about a phagocytic cell. And because it does require ATP, it is a form of active transport. So active transport is moving molecules from an area of lower concentration to areas of higher concentration, and it does require ATP. So with microorganisms, they do require specific nutrients in order to produce energy, to grow and divide. They also require very specific environmental factors, such as temperature, pH, oxygen level, all different types of things. So one of the first things that we'll focus on are temperature. So with bacteria, they all have a minimum, maximum, and optimum temperature range. And these terms are fairly self-explanatory, but the minimum temperature is the lowest temperature at which the microorganism can still grow and divide. If you go any lower than the minimum temperature, then growth stops. So typically if I'm working in the lab and I want to preserve cultures to look at next week, what I'll do is I'll take them out of the incubator and put them in the refrigerator. That drops the temperature below their minimum temperature so that growth stops and the plates are preserved for the next week when we need to look at them. There is also a maximum temperature. Maximum temperature is the highest temperature at which a bacterial cell can still grow and divide. If you go any higher than the maximum temperature, cell death usually occurs. So a lot of times if I'm working with bacteria, I want to know what their maximum temperature is so that when I go to sterilize the petri dishes in the autoclave, I have to make sure our autoclave gets above the bacteria's maximum temperature if I want to effectively kill them. But the maximum temperature is the highest temperature at which they can still grow and divide. Somewhere in between the minimum and maximum temperature, all bacteria have an optimum temperature, which is their preferred temperature. This is the temperature at which they would grow and divide at their fastest rate. So this is the temperature that they would prefer. Most, not all, but most bacteria tend to fall in an optimum temperature range of about 20 to 40 degrees Celsius. Most tend to prefer around 37 degrees Celsius, which is comparable to our internal body temperature. But optimum temperature is the temperature they prefer to grow and divide. They will, at this temperature, grow and divide at the fastest rate. So with bacteria, we can classify them based on what temperature they prefer to grow in, which is their optimum temperature. So again, we'll kind of go out of order. Most microorganisms are what we call mesophiles, meaning that they prefer to live in an optimum temperature range between 20 to 40 degrees Celsius. Again, our internal body temperature is 37 degrees Celsius, so any type of microorganism that can cause disease in human would likely be considered a mesophile because it would prefer this temperature in order to thrive. So that is consistent with most microorganisms, but there are definitely others. Microorganisms that tend to prefer a colder environment are called psychrophiles. So these microorganisms would grow best at temperatures below 15 degrees Celsius. So anything that would grow well in like the refrigerator or freezer, they would be considered psychrophiles. So for example, Staphylococcus aureus and Listeria, these bacteria are known for contaminating things like deli meats that you would typically store in the refrigerator. While the refrigerator would slow bacterial growth for most bacteria, with those specific bacteria, they prefer the cold temperature, so growth actually speeds up. So they are unique in the sense that they prefer a cold environment and are actually considered psychrophiles. Thermophiles are microorganisms they prefer a hot environment, greater than 45 degrees Celsius. So these would be microorganisms that grow well when exposed to direct sunlight. And then they're rare, but extreme thermophiles do exist. Extreme thermophiles grow best between 80 to 121 degrees Celsius. These types of microorganisms are often found in soil and water associated with volcanic activity. So they're definitely rare and not something we would consistently work with in lab, but they do exist. So it is important to know how we would classify bacteria based on what temperature they prefer. Again, if they prefer a cold environment, they're psychrophilic. A moderately warm environment, they would be mesophiles, and this is where most microorganisms fall. A hot environment would be a thermophile, and an extremely hot environment, those microorganisms would be extreme thermophiles. We also classify microorganisms based on their oxygen preference. So some bacteria prefer oxygen, some don't. Some don't care, they can survive in either type of environment, and then some only want a small concentration of oxygen. So we can classify bacteria accordingly based on that. If a bacterium is not dependent on oxygen, so they don't need oxygen to survive, we would classify them as being anaerobic. And if you put bacteria that are anaerobic in a nutrient broth test tube, those bacteria are going to grow at the bottom where they are not exposed to oxygen at all. They do not require oxygen to survive, so they have no need to intake oxygen. So they would grow at the very bottom. So that would be true for anaerobic bacteria. Then there's aerobic microorganisms. Aerobic microorganisms must have oxygen in order to survive. So they are very much dependent on oxygen. So if you put them in a broth test tube, they would always grow right at the top surface so that they would get direct exposure to oxygen for cellular growth. Then there are microaerophiles. Microaerophiles require a small concentration of oxygen. So they cannot sustain full atmospheric oxygen. Since they require a small concentration, they will typically grow beneath the initial surface within the broth so that they can get a small concentration of oxygen, but not pure exposure. And then there's the facultative anaerobes. These ones are the easiest to work with because they can grow in any type of environment. If oxygen's present, they'll use it. If oxygen's not present, they can survive without it. So in a test tube, most of them would grow at the top because if oxygen's present, they will use it. It makes it easier for them to generate energy. But if oxygen's not present, they can still survive without it, so you'll inevitably see some growing through that media, even towards the bottom of the media as well. So these ones are kind of neutral, and they can survive in the presence and absence of oxygen. So why is it that some bacteria are able to survive in oxygen, which we would classify as aerobic, and some bacteria are not able to survive in the presence of oxygen, which we would classify as anaerobic? It all comes down to what enzymes they have. Basically, aerobic bacteria are able to use oxygen because they have the enzymes to process it, and those two enzymes are superoxide dismutase and then catalase. Anaerobic bacteria are not able to use oxygen, and the reason for that is they don't have those enzymes, so they're not able to process it. So to kind of go through the process, when a bacterial cell takes in oxygen as an essential nutrient, the oxygen initially becomes toxic, and we actually call it a toxic superoxide ion. So as soon as the bacterial cell takes in oxygen, it becomes that toxic superoxide ion. If the bacteria is aerobic and able to use oxygen, that's okay. It's going to be able to break it down. So the first step, it will use superoxide dismutase to break down the toxic superoxide ion into hydrogen peroxide and then just normal oxygen that the cell can use. So that's kind of step one. Now the cell really doesn't want to have hydrogen peroxide, so it needs to break that down. So step two is to take the hydrogen peroxide that was made in the previous step and break down that. And that is broken down with the second enzyme, which is called the catalase enzyme. Catalase will break down hydrogen peroxide into water and oxygen, both usable materials that the cell can obviously use for things like ATP synthesis. So aerobic bacteria are able to utilize oxygen because they have the enzymes to break down the toxic superoxide ion into usable forms. Anaerobic bacteria do not have these enzymes. So if anaerobic bacteria took in oxygen and it became the toxic superoxide ion, they would die. They have no enzymes to process it and break it down into usable forms. So for that reason, anaerobic bacteria tend to stay far away from oxygen because they know if they take it in, they will not be able to process it. Whereas aerobic bacteria can use oxygen as much as they want because they're able to process it with superoxide dismutase and catalase. We can also classify bacteria based on their pH preference. So some bacteria prefer a more acidic environment, whereas others prefer a more neutral environment. If a bacteria prefers an acidic environment, meaning lower on the pH scale, then those bacteria are classified as acidophiles. So for example, Helicobacter pylori that can cause gastric ulcers in the stomach, that would be an acidophile. That type of bacterium would be able to thrive in the high acid content of our stomach with the gastric juices. Neutrophiles are bacteria that prefer a neutral pH, and this is most human pathogens because most of our body outside of areas like the skin and stomach have a neutral pH because our body is consistently made up of water, which has a neutral pH. So most microorganisms are around that neutral pH. They prefer around a neutral pH range such as like 7 as far as their pH preference. And then alkaphiles actually prefer to live in an environment that has a more basic pH, so higher on that pH scale. So definitely you don't have to know specific pH ranges because they tend to fluctuate, but you should know in general if a microorganism prefers an acidic environment, we would classify them as acidophiles. If they prefer a neutral environment, we would classify them as neutrophiles. If they prefer a more basic environment, higher on the pH scale, then we would classify those ones as alkaphiles.